So welcome to the second part of the muscle physiology PowerPoint. This is the part where we're going to get into the nitty-gritty of the anatomy of actin and myosin, and once we get the anatomy down, then we can go and talk about the physiology, meaning how each of those anatomical pieces works. So we're going to first start talking about the thin filament, and remember the thin filament is actin, and actin is a protein in and of itself, but in the skeletal muscle, the actin protein is made up of 4 different components, like subcomponents. First off, we'll talk about these red and yellow circles. These individual circles are called G-actin individually, globular actin. The G-actin comes together to make a long strand called F-actin, filamentous actin. There are 2 filamentous actins that are intertwined with one another. Now, each individual G-actin has a red portion and a yellow portion. The yellow portion is the active site. This is where you're going to do binding of other proteins like the myosin to attach them together and generate the muscle contraction. Right now, though, in this illustration, you see that all of the yellow binding sites are blocked by this green rope-like structure. This green rope-like structure is our next protein called tropomyosin. So tropomyosin is a rope-like structure that sits on top of and blocks the active binding sites on actin. In between the F-actins, you see right here in the middle, it's like a glue holding the 2 F-actin together called nebulin. So nebulin is found in between the 2 F-actins, gluing them together. And then on top of the yellow binding sites, blocking them is tropomyosin. And on top of that, you have these little orange nuggets called troponin. And troponin is there to bind to calcium. So we'll talk about that more in the physiology portion of this lecture, but you do need troponin, tropomyosin to do the muscle contraction, and both of these guys take part in the interaction with calcium. So the last thing I want to mention here is that these troponins are the subunit that actually bind to calcium. And when we again talk about the physio of this all, you'll see that calcium, when it's released, binds to troponin. That causes a structural change in troponin, which then, like a domino effect, will then cause a structural change in tropomyosin. And once the tropomyosin changes, it'll expose the active sites. Now, kind of moving on to the thick filament, the myosin, you notice that the myosin is covered by these little nubs. Each of those nubs is called a myosin head. And myosin heads are what actually bind to the yellow binding sites. So with that being said, if we see the structure as a whole, you have the actins on either side of the myosins, and the myosin heads sticking out on both sides to grab on to any exposed binding sites that you may have. Now, if you take a really close look, you'll notice that the myosin is composed of those nubs, but each individual nub does have a tail on it, and these nubs are called myosin heads. They have the ability to cock forward and back, so they can move back and forth like an elbow through this hinge over here. So the tail portion of it, they intertwine together to make the bulk of the myosin, and then the heads stick out to be able to bind to the binding sites on the actin. When you do have binding of the myosin head to actin, that's called forming a cross bridge. So that's a good term to know. Cross bridge formation. It's when the myosin head binds to the actin. Now, to touch again on how the contraction actually takes place, we have a theory called the sliding filament theory. And the sliding filament theory is simply the theory that you have Z line and Z line connected to the actins. And during a muscle contraction, what essentially happens is the Z lines with the actins slide in towards the M line. So you have that sliding filament theory, and if you recall, I had you circle the I band and the H zone. I had you circle those two because if you noticed, the I band and the H zone, those are the two that get substantially shorter during a muscle contraction based on the sliding filament theory. This is where the physiology begins. So if you need to pause the video and go over those structures of actin and myosin, once again, pause it now and then restart it once you're familiar with those structures, because I'm going to use those terms as common language in the explanation of the contraction physiology and the neuromuscular junction physiology. We'll start with the neuromuscular junction. Just based on the name, you know that this involves a neuron and a muscle. This is essentially where your neuron comes and sends a signal to the muscle itself. So let's say your brain, that's a brain, well, should look somewhat like a brain, sends a signal for the neuron to transmit the action potential to the muscle to tell the muscle to contract. What exactly is happening here? I do not need you to know the physiology of an action potential, but I do need you to know how we transmit that voltage down into the sarcolemma. So this will be the picture that we refer to at the end of it, and this is just like an introductory cover photo, but we'll kind of go over it just slightly right now, and then I'll go into the details of it. So you have the synaptic boutons, the tips of the neurons, release acetylcholine onto the sarcolemma. The sarcolemma transmits the action potential down the T-tubule to the cisternae to release the calcium onto the sarcomere to cause the muscle contraction. Right now, I want to teach you about this portion of it. The portion that has to do with transmitting the action potential from the neuron into the muscle. So this whole process is called an excitation-coupling contraction. It is the interaction between the neuron and the sarcolemma, and it eventually transmits that signal through the sarcolemma, down the T-tubules, to the cisternae to release the calcium, and it is the release of this calcium that actually is going to be a trigger for a muscle contraction. The muscle contraction does require ATP. You'll see that in one of the last mechanisms we go through, and it will produce tension because tension is produced when you contract a muscle, whether that's an isometric contraction or an isotonic contraction, there is tension produced. So let's look at the whole innervation process. You have a nerve sending an action potential down to the tips of the nerve where the boutons are. If you look at one bouton, you'll notice that there's these little red dots at the ends of them. These are vesicles full of acetylcholine. When the voltage from the axon reaches the bouton, it's going to cause exocytosis of acetylcholine onto the sarcolemma. So far, we haven't really talked about a lot of the mechanism. I've only mentioned that we're going to be talking about the action potential reaching the synaptic terminals, releasing the acetylcholine onto the sarcolemma. This is your mechanism slide. Make sure you go over this slide here. It's kind of drawn backwards. I don't know why the publisher did this, but we start at step 1 and we're going to run around to step 5. In blue, you have your synaptic terminal. You have the vesicles full of acetylcholine. In yellow, you have your sarcoplasm, and this blue lining is the sarcolemma. So here's the muscle, here's the neuron. When an action potential, and I'll just draw a neuron going this way, when an action potential reaches the synaptic terminal, that action potential causes these vesicles to bind to the membrane of the neuron and do exocytosis of the acetylcholine. So far, the action potential reached the tip of the neuron, causing exocytosis of the acetylcholine. Acetylcholine will then go and bind to the acetylcholine receptors. So in red, right there, is the ACH, and in purple, we call it the ACHR. R stands for receptor. So the acetylcholines will come and bind to these receptors that are found in the sarcolemma. Now, what it does that's super cool is that when acetylcholine binds to the acetylcholine receptor, it opens up sodium channels in the membrane. So binding of acetylcholine to the acetylcholine receptor in and of itself is the signal to open up sodium channels. Now remember, in any cell, where do you find a high concentration of sodium? Inside the cell or outside the cell? Outside the cell. So if you have high sodium outside the cell and you open a door for sodium, is the sodium from inside the cell going to want to leave the cell, or is the sodium outside the cell going to want to enter the cell? Per diffusion, sodium is going to want to enter the cell. So if you see these arrows, it shows sodium entering the muscle cell. Now, what charge does sodium have on it? It has a positive charge. If the resting potential of a muscle cell is around -70, and you're allowing a ton of sodium with positive charge to rush into the cell, what is going to happen to the overall voltage inside of this cell? Well, because positive charge is entering, that -70 volts is going to increase. And the term we use to describe an increase in voltage is depolarization. So we said that the sarcolemma was special because when positive charge enters the cell, it depolarizes the cell. And that depolarization shoots all the way through the sarcolemma, down the T-tubules, and so on and so forth. Now, we're not going to go over that mechanism here on this slide, but just know that that is the big picture of all this. When sodium enters, it causes the -70 millivolts to rise up to like -20. And that will send a voltage throughout the entire sarcolemma, down the T-tubules, to the cisternae, to release the calcium. This is how a voltage charge is transmitted from neuron to muscle. A lot of students think that this voltage shoots down and jumps into the muscle cell. Voltage cannot jump from one place to another in terms of your body compartments. It needs to be sent through a ligand binding to a receptor, open up the sodium channels, allow positive charge to rush in, changing the voltage of the cell as a whole. And when you change voltage of a cell, that is the generation of a new voltage signal in this cell. Now, do you want your acetylcholines to stay bound to the receptors for the rest of your life? Well, probably not. So we have these yellow half-moon shaped enzymes, and they're termed ACHE, acetylcholine esterase. I've typed out that word for you in a coming slide, so don't worry about writing it down right now. But it is the enzyme that pulls off the acetylcholine and removes it from the receptor so that it no longer is binding. It will disintegrate it, or it can recycle it and put it back into the actual neuron. So this whole process is the process of what you see at the neuromuscular junction. Everything that I've gone over in that illustration is written in slide form here as well as here, and there's that word acetylcholine esterase for you. So go ahead and read over those two slides whenever you have a chance. So this kind of segues us into the next mechanism, and that's going to be the mechanism of transmitting the voltage through the sarcolemma, down the T-tubules, to release the calcium from the cisternae and actually cause the contraction. So With that being said, I think what's important to be mentioned is that so far we've covered calcium, we've covered the anatomical structures of the sarcomere, but we haven't really talked about ATP a whole lot yet. This is where I show you the ATP cycle. This is probably one of the hardest parts of this PowerPoint. It's usually where students get confused on the material, so I'm going to go through it as slow as possible. And before I go through the mechanism, I'm going to review over this structure. So I'm going to draw the zigzag line. Here's actin. And I'm going to draw it very basic to keep this super simple. So there's actin. And then myosin is going to be in the middle. And the myosin heads stick out and they bind to the actin. So this is as simple as we're going to get it right now. What we're looking at here in this mechanism is the binding and contraction cycle of the myosin heads. So it's when the myosin head actually binds onto the actin and pulls it forward towards the M line. So how do we do this? Well, we all know ATP provides energy. And ATP provides energy in the form of phosphate bonds. So what you see in this first illustration up here is that you have the myosin heads and they're loaded with ADP and a phosphate already. What this means is the ATP came to this myosin head and gave the myosin head the energy. So I can put like a little symbol around it to indicate that this is charged and this is charged up with energy. Your myosin heads have to be charged up with energy in order for this whole process to even begin. So let's look at the actin now. You have the F-actin. You have tropomyosin covering the binding sites, which means myosin head can't bind to it. And then you have troponin, which are these orange nuggets. Nothing is happening here in terms of a contraction because, again, the myosin head has nowhere to bind to. These binding sites are blocked by tropomyosin. But how do we unblock them? Well, this is where calcium comes into play. When you have the voltage traveling through the sarcolemma, down the T-tubule, across to the cisternae to release the calcium, this calcium's job is to bind to troponin to cause tropomyosin to push out of the way. When you move tropomyosin out of the way, you expose these yellow binding sites, and like a magnet, this charged up myosin head is going to want to bind. So we're going to go through that step by step several times. Step 1. Let's say you sent the voltage signal, you release the calcium. Here's that calcium. It's bound to troponin. You've already pushed tropomyosin out of the way and exposed all of these yellow binding sites. So all the yellow binding sites are now open. Because this myosin head is charged up, he's ready to go. And what that means is the minute a binding site on actin opens, it's gonna attach to it. It's almost like a magnet. It prefers to bind to the binding site. So he binds. Now you have formed a cross bridge. So you need two things to form a cross bridge. You need calcium to have bound to troponin to pull tropomyosin out of the way. And you need ATP to have charged up this myosin head to give it the energy that it needs to bind on to the binding site. Now, step 3 is here. You made the cross bridge, but now you got to pull it all forward towards the M line. Remember, there's a hinge right here that flexes like an elbow. What is going to cause the myosin head to go from this orientation to that orientation. So to be bound from the back and then slide forward and pull this guy forward. Well, it's the release of ADP. So step 3 is the release of ADP and phosphate from the myosin head causes the entire actin to pull forward. The last step is to detach the myosin head so that we can go back and do another pull. In order to detach a myosin head, you have to bind on a new ATP. So again, I'm going to go over this one more time. In order to get this myosin head to cock forward and pull all of that actin towards the M line, you have to release the ADP and phosphate. Once it's pulled forward, binding of the new ATP will cause the release of the myosin head, and it'll give it a chance to move back to its starting position, as you see here, and recharge that myosin head with more energy to bind to a new binding site. So long as you have calcium bound and available ATP, you will constantly be able to detach, recharge, rebind. Make sure you go over this mechanism a few times because there is a lot of detail here. And this is also something that I'm planning on going over during our Zoom session. So I found a really good video. It is a 3D animation of the entire muscle contraction physiology, and it includes the neuromuscular junction. It includes the propagation of voltage all the way to the cisternae. It includes the release of calcium, and it includes the ATP cycle in the myosin head. Go ahead and click this link and watch this video. This is a great resource for you guys. Now, at this point in the PowerPoint, I'm going to let you guys read through these slides on your own because there are no mechanisms on this, but I will go through and I will kind of point out what material is really heavily on the test. So the term rigor mortis, make sure you know that term. Know the difference between an isotonic and an isometric contraction and a little bit about their characteristics. So isotonic is actual shortening of the muscle and moving of an object. And here are the little characteristics of isotonic. Isometric is when you create the tension in the muscle, but you don't actually shorten the muscle or move any objects at the other end. A great example of this would be like you pushing on a wall. So you generate the tension in your muscle. Your muscle does become bulkier, but it doesn't shorten. None of your bones actually bend at any angles, and the wall does not actually move. It stays stationary, but you do still generate a tension there. In terms of the energy, we talked a lot about ATP. ATP is the energy that we use in the myosin head. ATP hydrolyzes into ADP and phosphate, storing the energy in the myosin head, and your bodies are always making new ATP in the mitochondria. Now, ATP stands for adenosine triphosphate, ADP stands for adenosine diphosphate. When you make ATP, it doesn't just float around your body. It has to be stored in storage units called creatine phosphate. Creatine phosphate binds to and almost holds on to extra ATP. And this is actually a workout supplement that you can take to be able to store more energy before your workout. Then you have this enzyme called creatine phosphokinase, CPK. CPK is an enzyme, and it has the word kinase in there. So what do you know this enzyme does if the word kinase is in there? Well, kinases add phosphate groups to things. And wouldn't it make sense when turning ADP into ATP, really all you did was added the third phosphate group. This is also an ingredient that you can take that's part of pre-workouts. It's part of workout supplements that people who are like bodybuilders take constantly. Now, naturally, your body can generate ATP in the aerobic mechanism or the anaerobic mechanism. This uses oxygen. This one does not use oxygen. Make sure to read through aerobic and anaerobic and be able to differentiate between them, please. And then read a little bit about energy use. At peak usage, this means you're like maybe sprinting for an hour. And then this is where I want you to focus a little bit of your time. On the test, I've asked you to differentiate between the different types of muscle fibers we have. We have 3 different types of muscle fibers: fast fibers, slow fibers, and intermediate fibers. As humans, this is the kind that we have, intermediate. Chickens, turkeys, animals like that have 2 different types of fibers, fast and slow. If you recall, when you have chicken, you have dark meat and light meat. Some people are fans of light meat, like chicken breast. Some people are fans of dark meat, like the chicken thigh. What makes them different colors and different contents of like nutrients? Well, it's because they're different fiber types. Fast fibers, they're called fast because they contract very quickly. These fibers can contract quickly, but they also get tired really fast. They don't have many mitochondria, and without mitochondria, what can you not make? You can't make a whole lot of ATP. These guys are also called fast glycolytic fibers rather than oxidative fibers because they do anaerobic respiration. They do not utilize oxygen. Now, slow fibers are slow to contract, but they can go for a long time. Why? Because they have a lot of mitochondria that can constantly make ATP over a long period of time. These guys also have myoglobin in them. Myoglobin is like hemoglobin, and it holds on to oxygen in your muscles. Because it holds on to oxygen, we call these slow oxidative fibers. Now, the color of this is also red, like a dark red color. So because of that, your slow fibers is your dark meat in the chicken. This is your chicken thigh. The fast fibers is your light meat. This is like your chicken breast. In terms of what they look like, slow fibers are going to be, again, darker because there's more myoglobin, and fast fibers are lighter, but nonetheless, they both have striations. So this is just the chicken example that I gave. In humans, we have pink fibers. It's not dark or light meat in humans. That's because we have a mix of both of these. We get the best of both worlds. We're able to pick up fast, like the fast fibers here, and we're able to go for a long time, like the oxidative fibers here. So be sure you know the difference between these 3 fiber types. I think I made this a matching section on your test. So I gave you a description, and then you have to match it with which fiber type it is. So with that being said, you've completed the 2nd video of the muscle physiology lecture. Go ahead and let me know if you have any questions, and I'll talk to you guys soon. Thank you.